right, these are your notes on genetics, chapters 13, 14, and 15. I'm going to scroll over so you can see all of my notes. Mendel's laws. Mendel the monk studied pea plants. He looked at monohybrid crosses first, which are single gene traits on single chromosomes. So you have two alleles, you like either a tall or a short or a red or a black. And these results led to the law of segregation. In the law of segregation, it says that diploid cells have pairs of genes on homologous chromosomes. The two genes of a pair will separate from each other during meiosis, so they end up on different gametes. So my lovely little picture here, you have, I've already started annotating because I did this video once before, but you have chromosome number 1 from dad and number 1 from mom. You have chromosome number 2 from dad and number 2 from mom. So this would be in G1. Then in S phase, they're going to synthesize. So each one of them will make a copy. And then in G2, you end up with twice the number of chromosomes. So you have number 2 here from dad and 2 here from dad and 2 here from mom and 2 here from mom and so on. They are sister chromatids, so they're attached at a centromere. One of the questions you guys missed on your test was worded like this. It says, how did the daughter cells at the end of mitosis and cytokinesis compare with the parent cell when it was in G2 of the cell cycle? Well, G2 of the cell cycle looks like this, which means you have twice the number of chromosomes as normal and twice the number of DNA as normal. And so the correct answer would have been the daughter cells have half the number of chromosomes and half the amount of DNA at the end of mitosis. So I just wanted to make a note of why pretty much everybody in the class missed that question. The next thing I want to mention is that after you do those divisions, then these homologous chromosomes will split. So in meiosis 1, you have chromosome number 1 from mom and chromosome number 1 from dad line up like this. And so when they split down the middle, chromosome number 1 from mom will end up on one of these cells and chromosome number 1 from dad ends up on the other cell. Now you notice that they're slightly different colored, which we'll talk about here in a second. But when they split again, you end up with half the number of chromosomes total. So when he studied dihybrid crosses, di means two. So when he studied two hybrid crosses or two trait crosses, this led to the law of independent assortment. In an independent assortment, paired genes on homologous chromosomes tend to sort into, let's bring up my notes so I make sure I tell you right, into gametes independently of other gene pairs during meiosis. So whatever's on chromosome number 1 can line up however it wants and end up in whichever gamete that it chooses to at the end of meiosis, independently of whatever's on number 2 or 3 or so on. And this is only a mess if they are not linked. So if genes are linked, that means they're close enough on the chromosomes to where they'll be inherited together. So we've got things like a gene that would be here and a gene that would be here. Those would be considered linked because they're not going to be able to split off usually if they're really close together on the chromosome. But if you're not linked, and let's say you have a gene on this end and a gene on this end of the chromosome, then you could be inherited independently of each other because this is non-linked genes. So if crossing over occurs and some of mom's chromosome gets on dad's and dad's on mom, the recombinant chromatids now would look different. And so on non-linked genes, you can mess up those laws of independent assortment. So it says here, crossovers can break up linkage groups. The next pattern we're going to talk about is autosomal recessive pattern. This is a trait that, oh wait, sorry, I lied. Don't write recessive, write dominant. Autosomal dominant pattern comes first. This trait appears on everyone who carries it. So it happens in both sexes with equal frequency, and it appears in every generation. So whether you are big B, or big B, little b, either one of those, you would still exhibit this dominant trait. However, in opposite of that, in an autosomal recessive pattern, this trait specifies that it appears only in homozygous people. So you would have to have 2 little chromosomes in order for that to happen. But it will also occur equally in both generations. Due to its recessive pattern, meaning you have to have both recessive alleles in order for the gene to be displayed, it can skip generations. So you might see it in great-grandparents and then maybe even again in their great-grandchildren. So it can skip around. Other patterns of inheritance, some of these were found by Mendel, but others were discovered by other scientists. The first one I want to discuss is incomplete dominance. During incomplete dominance, the phenotype of heterozygous individuals is an intermediate blend of two homozygous phenotypes. So it's blending. So think of paints. In my beautiful little picture here, we have red paint and we have white painted flowers. When those mix and you end up with heterozygous individuals, you end up with pink flowers. So it blends one from the other. The next one is codominant. In codominant traits, heterozygous individuals will have both homozygous phenotypes. So this may use a multiple allele system, such as blood typing or having ABO. In my beautiful picture, you've got a black bear and a white bear, and you end up with what looks like a panda, which is not a bear, but it's exhibiting both the dark and the white phenotypes. In your ABO blood system, A is dominant, and B is also dominant, but O is recessive. So to be type A, you could have AA, or you could be AO, but the A then is dominant and it masks. To be type B, you would be BB or BO. To be type O, the only way you could be it is OO, and so you have neither dominant phenotype. But the codominance part of this is you could have type AB blood, which is the most rare form of blood, but that means you have the A allele and the B allele. So you exhibit the antibodies in your blood of type A and you have neither antibody type A or type B, but you have both antigens A and B exhibited on your blood cells. So you would have A and B antigens on your blood cells. So you have neither A or B antibody. So that is a unique way of inheritance, and it affects who can give blood to whom and how that works. So if you're interested in that, look into it a little bit farther on your own. I'm going to keep going with these other notes because you should have had that in regular biology. Type C is epistasis. In epistasis, you have two or more genes that affect the same trait, such as pigments in skin and in fur. So in my beautiful picture here of mice, we have two traits that are affecting the same thing. So we have a trait B and a trait C that can affect fur color. And you see all the different combinations that cause dark. In all of these, there's at least one capital B and one capital C. In the ones that are brown, you have lowercase b's and capital C's. And the ones that are white, however, all you need is two little c's, and you can have two little b's or two big B's or a heterozygous for B's. But as long as you have two little C's, you're going to be without pigmentation. You're going to be an albino mouse. So that's kind of unique to the mouse world that isn't in other things. But the idea is, in this epistasis pattern, is multiple genes will affect the same thing. In this case, we have two genes affecting coat color. The next one we're going to discuss is pleiotropic individual or pleiotropic genes. These are genes which affect two or more traits. So it's one gene that can affect multiple traits. In my example there, it's saying Marfan syndrome, which you can look up. But in this one, this picture that I was able to find, it's got this beta globin gene mutation, and this beta globin through pleiotropy affects eyes, so it can cause blindness of the eyes. It could cause liver failure, and it could also cause heart attacks. But what happens in this beta globin mutation is, instead of having a beautiful biconcave disc shaped to your red blood cells, you end up with sickle cells or sickle cell anemia. So sickle cell anemia can cause all kinds of other trait issues, not simply just the shape of your red blood cells and your ability to carry oxygen in those. Then you have continuous variation. Now, in continuous variation, you have a variation that can have different little changes along the line. So it says a range of small increments of phenotype due to multiple gene influences. So this generally makes a bell-shaped curve. You can see a beautiful bell-shaped curve here in this human height diagram. It comes up and comes back down. So you're going to have some on one end of the range, some on the other end of the range, but you're going to have a lot of individuals in the middle. And here's one in seashell color. You have some that are really dark and some that are lighter, but you have a lot of them in the middle having this more common seashell color. And what causes this is it is in short tandem repeats. So they spontaneously can expand or contract quickly, creating multiple alleles. For example, longer repeats are longer doggy faces. So they have found that there are 12 alleles that affect the length of a dog's face. And those all have to do with how many short tandem repeats there are. If you have 12 repeats, then you are going to have a long doggy face, long doggy face. If you have much less, like 1 or 2 repeats, you're going to have a very short doggy snout, like a pug. So depending on the number of repeats that are there, you're going to have your bell-shaped curve where most of them are in the middle, but you're going to have some really long snouts and some really short snouts, depending on how many repeats are available there. In X pattern inheritance, you have genes that are carried on your X chromosome. So it is a recessive pattern. This is because the X has different genetic information than the Y chromosome. They used to be homologous chromosomes, but over time, and we'll talk about the mutation that caused that later, but over time they changed. So the X's information is totally different than the Y information. And so let's say that we have this combination of individuals. We have an unaffected father, but a mother who's a carrier. So phenotypically, she's normal, but she has the trait on her X chromosome. And let's say these two breed and have children. They might have an unaffected son if he has a normal X from both mom and dad. If he inherits a normal X from mom and the Y from the dad, I skipped over here. You could have an unaffected daughter if you get a normal X from mom and dad, because remember, the dad can only give the normal X. You could have a carrier daughter if dad gives normal X, but mom gives this recessive carried trait, which is whatever this is. This could be hemophilia, for example, or color blindness. And you could also, from this set of parents, have an affected son. So dad would give the Y and mom would give the affected X. So what's going on here is you have 4 different genotypes, but honestly you have 2 different phenotypes, the unaffected and the affected. And that matters because you could have a carrier daughter and have a line of carriers and have it not be expressed until much later down the line, depending on how many children you have or if you have sons or daughters. So to finish our notes, so it's more often found in males. Carrier women have the dominant normal allele that matches the recessive allele, and males can pass on the trait to daughters, but they can't pass it on to sons. So in the next video, I'll go over the next set of notes.